Hello, I'm Cece, and my group mates and I, Leanna and Nina, will explain to you the phenomena of the central dogma of biology. This can be explained with DNA replication, transcription, and translation. We will further go into more detail.
Replication is the first part of the central dogma. In this process, the DNA strand gets unzipped with help from an enzyme called helicase. Helicase breaks hydrogen bonds that are holding the complementary bases together. Once the strands are separated, those two strands will then be used as a template strand or a model to help make the new strand. The name of the two strands are leading and lagging. Leading strand goes from 5' to 3', and the lagging strand goes from 3' to 5'. The leading strand is continuous and the lagging strand is discontinuous. The nucleotide base adenine will always pair with thymine, and guanine will always pair with cytosine. For the leading strand, there is an enzyme called primase mix primer that binds to the end of the leading strand and begins DNA synthesis. After the primer is set at the end of the leading strand, DNA polymerase comes in and begins to read the strand and adds new complementary nucleotides from 5' to 3'. For the lagging strand, there is an enzyme called primase, makes multiple primers, and they begin to set at different points on the strand. After the primers are set, DNA polymerase comes and does one segment and then finds another primer and adds complementary bases in that segment, which are called Okazaki fragments, from 5' to 3'. To make a continuous strand, exonuclease will remove primers and DNA ligase will fill in the gaps. The end product of replication are two identical copies of DNA. Each one contains a template strand, colored, and a new strand, gray. This is called semi-conservative model.
Transcription is the second part of the central dogma. In this process, the DNA gets transcribed into RNA. DNA cannot leave the nucleus, so in order for the information to get copied, it has to be transformed into RNA.
For transcription, there should be one segment of gray, but we have used both segments as colored.
Before we can start the transcription process, the DNA has to unwind near the gene that is going to be transcribed. This region that gets unwinded is called the transcription bubble. The first step of transcription is called initiation. In initiation, there is a certain part of the DNA that has what we call a Tata box. The Tata box is just a segment of DNA full of thymine and adenine, which are two nucleotide bases. During this stage, transcription factors will bind to the promoter region. Those factors are transcription factor 2D, H, E, F, A, and B. All of these bind to the promoter region or the transcription bubble until RNA polymerase were to unwind and start transcribing. The RNA polymerase has high affinity for the promoter region, which is the Tata box or transcription bubble. The next step in transcription is elongation. Once it attaches, RNA polymerase goes along the template strand, synthesizing or making an mRNA strand. The RNA polymerase reads the template strand from 5' to 3' in order to make it 3' to 5'.
It's important to remember that adenines for mRNA will pair with uracil instead of thymine. DNA recombines after the bases for mRNA are made.
Lastly, we have termination. Scientists haven't had too much information on this part of transcription, but we have a basic gist. Basically, the RNA polymerase gets a signal to stop transcribing. In order to protect the mRNA, there is a 5' cap that is added to the 5' end, adding 3 phosphates. Also, there is a poly A tail on the 3' end, which adds adenines to the side. These are added in order to protect the mRNA sequence. After this, the mRNA leaves the nucleus and goes to the ribosome.
Translation is the last part of the central dogma. During this process, all three types of RNA is used. The mRNA will make a protein. Once again, there are 3 steps in the translation process, which are initiation, elongation, and termination. The mRNA, which was made in translation, leaves the nucleus from nuclear pores and will enter the cytoplasm of the cell. From here, it makes its way to the ribosome, where protein synthesis will occur. In the inside of a ribosome, you will find an A site, a P site, and an E site. Each one can hold 1 tRNA at a time and have different tasks. Initiation is the first part of translation. In the small ribosomal subunit, mRNA is read by its codons. Every 3 nitrogen bases is called a codon, and every codon will code for a specific amino acid. The order of the nitrogen bases ultimately gives the order of amino acids which will make our proteins. There are different tRNAs or transfer RNAs, which have a very important role in translation. These molecules have something called an anticodon on the bottom that is complementary to our codons found on mRNA. On the top of tRNA, it holds an amino acid that matches to the anticodon complementary codon. For the process of making a polypeptide chain to start initiation, the tRNA from methionine needs to find the start codon AUG. This codon starts the process. Elongation is after the start codon is found, and this process is the creation of a chain of amino acids that will bind. The tRNA will enter the ribosome, and the A site will bind and find its match by reading the mRNA codon. Once the anticodon found its match and is bound, it will move to the P site, where the amino acid at the top will detach and wait in its protein channel. The reason that the amino acids can separate from tRNA is because they are held together by a hydrogen bond, which is weak, so it breaks. When the A site is free, the next tRNA comes in. After the amino acid detaches from its tRNA, the tRNA will travel to the E site, where it gets ready to leave the ribosome. Because the P site is now open, the second tRNA will move along to the P site, and the amino acid will detach and bind to the previous amino acid that is sitting in the protein channel. Once again, the tRNA is in the P site, moves to the E site. This process continues until we have made a polypeptide chain, which are amino acids bound together by a peptide bond. Termination process occurs when a stop codon is read. The stop codon on mRNA can be UAA, UAG, or UGA. Once this is read, a molecule similar to tRNA will enter in the A site and stop the chain of amino acid. This chain is known as a protein. Once the amino acid chain or protein is completely made, it will leave the ribosome via the protein exit channel and will get ready to be sent off and perform its job throughout the body. However, once again, if there is a mutation or a change in the mRNA, everything will be affected. If nitrogen bases are substituted for another, that codon will code for a different amino acid, which will create a different protein or change its shape. This directly correlates with structure and function, because if the structure of the sequence to make a protein is changed, the function for that protein will also be altered.